I'd like to thank my colleagues at the EMA for hosting us. I think this is a wonderful opportunity, and it's very nice to see some faces from other regulatory agencies and other sectors sort of meeting on European soil. For the next 15, 20 minutes or so, I'm going to talk about sort of the recent activities at the US FDA in the area of pharmacogenomics, with particular emphasis on the guidance for early phase clinical studies. But I'll touch on some other areas as well. So I think we're all aware that pharmacogenetics, genomics is not a new concept, and in the U.S., the National Academy of Sciences, even 40 years ago, sort of advised that it was very critical to understand how genetics affects our ability to break down medications. So this is not a new concept. It's just now it's becoming very more commonly assessed in the context of drug development for new drugs, and there's increasing discussion about it in the context of clinical practice. So while the science is not necessarily new, our technologies and infrastructure to apply this scientific information has changed quite a bit to basically foster the science. So for those that aren't familiar, the FDA is a very large agency, and we oversee many medical products and other foods and such consumed by folks in the United States. It's worth noting that each center in the United States has its own burgeoning genomics group or pharmacogenetics group. In the Center for Veterinary Medicine, they're very interested in this for looking at in animal studies. CDRH oversees diagnostics. The Center for Biologics deals a lot with vaccine health, and they have interest in it from that standpoint as well. So my home is in the Center for Drugs, so that's the perspective that I'm giving you today. And even within the Center for Drugs, there's a variety of groups that deal with pharmacogenomics, both in the Office of New Drugs, Office of Translational Sciences, and even in the post-marketing setting in the Office of Surveillance and Epidemiology. So within the Center for Drugs, there's going on about a decade of history with dealing with pharmacogenomics and sort of the regulatory science and policy around it. We started in 2005 with the voluntary data submission program. We also started a pilot program for biomarker qualification. We were very actively involved in labeling updates for approved therapeutic drugs. And we also issued guidance on how and when to submit genomic data to the agency as the science was developing. What we're seeing now, sort of in the past 10 years, is the voluntary submission program has changed and probably will continue to evolve. While the number of submissions hasn't changed, their character has a little bit. We're seeing more biomarkers that cover broad therapeutic areas, have potentially multiple uses, as well as very exploratory data, such as potentially things like next-gen sequencing. The biomarker qualification program is now an official process at the agency. Labeling updates haven't all been performed to this day for marketed drugs, but we're seeing a shift now to drugs that are new to the market. So we're seeing pre-marketing labeling rather than post-marketing labeling updates. And just to sort of illustrate that, this is a listing of some of the new molecular entities that were approved in fiscal year 2011. You can see that many of the molecules that were submitted to the agency had some type of genetic biomarker, some protein predictive biomarker within the submission. And many of these translated to labeling. So it's important to point that of the 35 new molecules that the US FDA approved, about a third of them had some type of genomic biomarker information in them. Some of this was voluntarily submitted or unsolicited submission. Many were also requested by the agency. So also with the maturation of the types of submissions we're seeing and the evolution of the science, we're also seeing maturation of our guidance and policy in this area. So as I mentioned, we started in 2005 with a guidance on data submissions and a concept paper on co-development. And we subsequently saw evolution in certain therapeutic areas. So for instance, in the setting of chronic hepatitis C, we have guidance to study specific genetic factors in the context of clinical trials, because it's something that we've discovered over the course of years that there's very important genetic markers in the setting of hepatitis C. We also have broader guidance, as I'll mention in the next couple of slides, on pre-marketing evaluation for pharmacogenomics. We also have a guidance on companion diagnostics because in the US we also regulate medical devices. And we also have guidances in the works for enrichment trial designs, which is very important for pharmacogenetics as well as co-development. So with the growth of pharmacogenetics, we've also seen a number of different companion diagnostics also approved in the past year. So we had a number of successes in 2011 and 2012 where companion diagnostic was approved with a drug. Those are listed here. Many of you are familiar with these. And this, I think, speaks to the increasing collaboration within the US FDA between the different centers that oversee these drugs and devices. So now that we're sort of physically housed in the same location, there's much more communication. We're present at all of the meetings together in the setting of new drugs being developed. So I think that that's doing a lot for the success and development of policy in this area. That's sort of the old news, and we'll talk a little bit about the clinical pharmacogenomics guidance. One concept that is probably important to introduce is what the concept of a problem drug is, and this is essentially how we think about it. So my comments here, most of them reflect what's actually written in the guidance. Some is also editorial based on sort of our practical experience in applying the principles of it. So problem drugs are generally what we're most concerned with. These are drugs that are highly variable, as was alluded to in the previous talks, drugs that have disproportional dose to exposure relationships or exposure to response relationships, situations where we see race effects, or where there's outliers or no monitoring tools, especially when it's a morbid or mortal condition. So these are the things that we actually attend to most. We don't give much attention to a topical product that has very low systemic exposure, for instance. Clearly, it's not important to bank DNA necessarily in that setting. So the guidance essentially spells out, when to consider how genetic variations affect, the drug's pharmacokinetics, efficacy, or safety, and it really just provides general principles. And it's important to note that this does not relate to later phase trials. It relates only to early phase clinical studies that are either observational or exploratory in nature. The issues around confirming pharmacogenetic effects in phase 3 trials is subject of co-development and enrichment guidances. So, this has been discussed before. One thing I will point out is that pharmacogenetics is a lot more than sort of a co-developed drug and diagnostic. It's really a tool that we have at our disposal to help understand variability in drug exposure and variability in drug response. So often these tools can be used to characterize mechanisms for adverse reactions, for instance, without a clear intent necessarily to develop a diagnostic that's used in the clinic to predict a response. It just helps us understand biology. That said, there's also situations where the potential outcome might be something a little bit more prescriptive, where you're selecting a patient based on the risk-benefit profile that's established in a genetic subgroup. You're modifying dosing, or you're intensifying some clinical monitoring in the setting of clinical trials, perhaps adverse event monitoring in the setting of clinical use, potentially just patient monitoring and follow-up. The genetic factors that we're most interested in are fairly obvious. It's the drug metabolizing enzymes, transporters, and the target. Often we also pay attention to disease markers, as these can be used to enrich clinical trial populations for prognostic purposes, and also have important role in understanding the risk-benefit of a drug. A drug's treatment effect versus placebo may be very different in someone who has a better prognosis based on whatever biomarker as compared to somebody who has more advanced disease. Ultimately, this is getting at PK and pharmacodynamic variability with the eventual effects on response and efficacy, or tolerability and safety. So the basic, foundation of the guidance is really to recommend DNA be collected throughout all phases of drug development and all clinical trials. That's sort of the default. Practically, we, as I mentioned before, apply this in a little bit more narrow of a scope. We usually look at things, if there's evidence of multimodal PK or there's a clear safety issue, particularly those that are idiosyncratic. We have some type of variable exposures or responses. or we've observed a race effect in clinical studies. Those are situations where DNA collection becomes more important. And we expect that it be collected in all subjects if there's a specific hypothesis to be tested. So if you know your drug's metabolized by 2D6, it behooves you to collect DNA in everyone so you can make the most out of the analysis of that particular genetic factor. If you don't have a hypothesis, but some of these other things apply, it's good to do it to the extent possible. I know we're dealing in a global drug development environment, so that consideration becomes very important. Also for rare events or adverse events, sometimes you can target collection, particularly from those patients who experience either some type of clinical toxicity or, if it's a phase 3 trial, the primary endpoint. And we also expect that if you're not collecting DNA from all subjects for any of these purposes, that there's some disclosure of what the reasoning behind that is, and often tying back to geographic differences. So other principles are that you obtain broad consent, that you collect samples before randomization in the setting of phase 3 trials to minimize the amount of bias, that samples be retained, as I mentioned, that we document reasons for incomplete sampling, and that there's some record of sort of the analytical and sample quality that goes into the storage and analysis of these specimens. So I won't spend too much time on this other than to sort of just reiterate that in certain settings we have very specific hypotheses, and those can be tested, on a one-off basis using, individual tests. In some situations, it may be better to use these higher throughput genotyping chips. For instance, if there's some unresolved PK variability that we don't quite understand, we haven't been able to identify anything in a non-clinical cell culture type of experiment, but there's still important variability that might affect the way the drug responses are. That's important. You could potentially use one of these higher throughput chips to get at that issue. And additionally, when selecting chips and selecting genetic markers for analysis, it's very important to consider the racial ethnic group that's being enrolled in the clinical trials and to whom it will ultimately translate. Also, in terms of the differences between retrospective genotyping and prospective genotyping, retrospective studies are generally fine for PK using conventional population pharmacokinetic approaches, or for safety endpoints where it's more appropriate to do a case control type of analysis. There may be some instances where prospective genotyping is warranted. Clearly, if you're doing a clinical trial that you're trying to determine something prospectively, stratifying randomization or enriching, you have to do that up front. But also, you do this prospectively where you're trying to get a very thorough pharmacogenetic assessment, or you're excluding, for instance, poor metabolizers from a drug interaction study. So you're optimizing the chances of seeing that signal in its worst case scenario. So one example that I think is somewhat instructive is sort of in the potential use of this in early phase studies. We always hear that it's difficult because there's limited data. It's early phase, you haven't generated enough of a database for the molecule yet. But if we look at the IL-28B story with chronic hepatitis C, it's somewhat instructive. We saw that from retrospective sub-studies from a clinical trial, there was a very large signal for IL-28B being a predictor to the response to the old 2-drug regimen that was the standard of care for hepatitis C. Now, this is something that probably could have been detected in the context of a phase 2 trial. So in this earlier phase exploratory trial where you're understanding dose and responses. So it is conceivable that you could have conducted a genome-wide study and come up with something that was robust in that setting. Now here we had dozens of external experiments sort of validate the role of this genetic marker. In the context of drug development, we would be relying on things like in vitro studies and whatnot to sort of help substantiate any type of signal. But nonetheless, there was regulatory acceptance of this as a genetic marker. It was quickly adopted in guidance, as I mentioned before, and it's now a standard analysis factor for controlled trials in this area. And we evaluated some of this data in the context of new drug applications, and there's also some drug development programs that specifically target the less responsive genotypes to the standard of care. So a very interesting story and instructive from a clinical standpoint. So this essentially enumerates some of the same points. In early phase studies, you identify and discover a biomarker. You then proceed to confirm the biomarker effect as well as the treatment effect on a more robust endpoint, whether it be a surrogate that's widely accepted or a clinical outcome. And with this pathway, you can achieve either Subpart H accelerated approval in the United States, or if it's an outcome, you can do full approval. So the principle is that there's still development and policy being worked on, include enrichment of clinical trial populations, as well as companion diagnostics and co-development. So these are areas that you'll see more advice from us in the future on. I'll just say a couple words on the companion diagnostic guidance, because this is receiving a lot of attention. It's also something that's in stages of being near finalized. This document really defines companion diagnostics, and this is important in the United States because we regulate medical devices. And what we've come to define as a companion diagnostic is really an in vitro test that is used to, that are essential to the safe and effective use of the medication. And this can be whether they're predicting response or adjusting dosages. So in this document, we describe our policies, basically stating that we will regulate these devices before they come to market, and that is risk-based. So by saying risk-based, I mean if a drug is really important or a test is really important to making a critical treatment decision, whether or not a patient receives a therapy, those will be subject to FDA regulation. And also even in the pre-marketing setting, there will be some oversight of this because obviously entering a patient into a clinical trial has safety implications for the patient. So there needs to be some assessment of the analytical validity of those tests in that setting. Recently signed into law was the Prescription Drug User Fee Act, which reauthorizes this act. And in the text of this, there is some additional support that's going to be provided to pharmacogenomics and exploratory regulatory science and expediting drug development. So specifically related to biomarkers and pharmacogenetics, we're hoping to develop more internal capacity to review these very complex submissions. We're going to be training staff in this area, and we'll also be holding a public meeting in the next year or two on these topics. So with that, I'll just conclude basically by saying that we've attempted to sort of foster the use of genomics in drug development. It's very important, and over time we'll get more granularity on the issues that are becoming really challenging, which is how you confirm this effect and how you sort of adequately vet the diagnostic before coming to market. With that, I'll close.
Thank you very much, Michael. That was an excellent overview of what we experience with the FDA experience of pharmacogenomics and how you handle it. It's good to see that the companion diagnostic part is now becoming a part and parcel of the overall system. What I would like to say that we'd probably like to be in the same situation in Europe, but we're slightly different.